Hello everyone, welcome to my channel. In today's video, we are going to discuss about National Institute of Immunology previous year questions. Now, before discussing the important topics and the list of questions, we have to look at the exam pattern. So in this exam, you have to attempt around 60 questions in 90 minutes, and for each correct answer, you get 3 marks, and for every incorrect answer, you get -1 marks. So they have 1/3 negative marking. So attempt the questions really carefully because you tend to lose a lot of marks because of this pattern. Now, from the list of important topics, you have I'll mention a few of these topics which are really important, and if you are preparing for this exam, then just if you don't have the time to look at all these topics, at least prepare these 3 topics that I'm going to tell you. The first one is biochemistry, second is immunology, and the third is methods in biology. These 3 are a must for this exam because most of the questions are asked from different aspects of these 3 topics. So prepare these 3 topics really well. Other than that, they have questions from molecular biology, applied biology, animal physiology, especially the hormones part, and some questions from cell biology and signaling. Very little from signaling part, but cell biology they ask. Now moving on to the questions. So starting from biochemistry, you have to know about how to calculate the charge on a peptide at certain pH and pI calculation, because it is very frequently asked. They ask about pI calculation, charge calculation, charged amino acid they can ask to calculate its pI, so that you should also know. They asked about vitamin D precursor, so along with this you should also know about all the vitamins and the cofactors that they donate, and also about the precursor of NADH in Krebs cycle. So again, Krebs cycle, glycolysis, etc., that all you should know. Amino acids and their structural feature, this is one of the most important topics because a lot of questions can be asked from this area, including the chiral centers, which amino acids act as helix breakers, the amino groups, imino groups, or whatever the aromatic groups their name is, that all those questions can be framed from this topic. So very important topic. They asked a question from rate constant of an enzyme and half-life calculation, malate aspartate shuttle. Basically, you'll have to study both the shuttles and what molecules are involved in this shuttle. They can ask questions like which molecule is involved in malate aspartate shuttle, and they will give you examples, some of the options, and you'll have to select from that option. So you should know about the components of every I'll say process that you're studying, be it glycolysis, Krebs cycle, shuttles, electron transport chain. You should know the name of all the complexes and everything, even the sequence in which these things work, that you should know. You have to study biochemistry really well because a lot of questions can be made from that area. They asked about short chain fatty acid. Basically, these are produced by the gut bacteria. There are 3 major SCFAs, that is acetate, butyrate, and propionate. Then again, important, they have repeated this topic many times. You should know about this endopeptidases as well as exopeptidases. Chymotrypsin is their favorite topic. Also, you should know that what lies at the active side of chymotrypsin. Again, I'm saying that whatever topics that I'm mentioning, try to read from all the related areas from these topics. They asked myoglobin's position in Ramachandran plot. So read about myoglobin, what is its structural feature, and based on that, where will it lie in the Ramachandran plot. So it is made up of alpha helices, again right-handed alpha helices, so it will lie in the lower right quadrant. You should know about these basics. So again, etc, effect of replacing magnesium with manganese, this you should know. Basically they have no effect because their structure is quite similar. This will be Mg2 plus Mn2 plus. Okay, Now, enzymes involved in glycolysis, gluconeogenesis with opposite functions. Again, you should know about all these cycles, all these basically steps you should know. Then enzyme kinetics, again very important. Many calculations are asked from this area. Also the Lin-Weaver-Burke plot, enzyme inhibition, Dixon plot, everything you should know. Enzyme regulation also. And in this Lin-Weaver-Burke plot, basically they can give you a graph and can ask that if an inhibitor is added, what do you think will happen to these lines? Will they move parallelly or anti-parallelly? Then again, solution related questions were asked. There are 2-3 questions were asked. They can be about molarity or normality or molality, so that you should know. Now moving on to molecular biology questions. They asked about RNA induced silencing complex. So to read about this, dicer, drosha, snRNA, snORNA, miRNA, siRNA, all these related topic you should know. Now, effect of mutated TBP on which polymerase? So if you want to know the basic process of transcription, you wouldn't know which polymerases are involved or which molecules are involved, data binding proteins, like what they do. You should have idea about all these steps, then only you will be able to solve these questions. Then they asked a question about euchromatin and heterochromatin and like how they differ, and from the given options, H3K9 methylation was the answer. Along with this post-translational modification you should know, different post-translational modification that happens, and you should have a rough idea about the molecular weight of the effect of molecular weight on of these modifications, because some modification they are heavier and some are lighter. So the question might ask that a protein after post translational modification showed increase in its molecular weight by something Dalton or whatever. So you should have a rough idea that which are like what these compounds are made up of, like which are added in post translational modification. Then bacterial replication and enzymes involved in its regulation, lambda fast gene regulation, attenuation in TRP operon. Along with this, read about catabolite repression, glucose effect, and also about lac operon and cap protein, everything like related topics, everything you have to read. Then alpha amyloidine affects which polymerase activity, also the sequence in which it affects polymerase 2 the most, and then if the concentration is increased it affects polymerase 3 and it has no effect on polymerase 1. Also what these polymerase they transcribe. So you should know that apart from the basic tRNA, mRNA or rRNA, the other non-coding RNA that are transcribed from these polymerases you should know, because a lot of questions are being asked from RNA biology, so you should have some idea about that. They asked about translational fidelity, like how is it determined. So amino acyl tRNA synthesis, they basically are responsible for this. Glycogen signaling, they asked like which signaling pathway it takes place, like what molecule is responsible for this. So this is GPCR pathway is involved for glycogen signaling. Then effect of mutant tap protein, function of cytoskeleton, integrin connects which structure, this is cell matrix, channel responsible for action potential. Then they asked about the function of peroxisome, mutant RAN gap effect. Again, along with RAN, read about RAS, Rho, Reb, and all those monomeric GTPAs which are involved in different pathways. You should know what they do and where they are involved. Now there was one question from cell fusion experiment by Rao and Johansson. Like for example, you have two cells, one is in G2 phase, the another one is N phase. If they fuse, what will be the phase of the resultant cell? So see this topic, it's from cell biology, and you can get this topic from, I think, Lodish you can get. Then they asked about myasthenia gravis autoantibody. So make a list of all the autoimmune diseases and the auto antibodies for those diseases, because it is like they can be asked. Cells involved in acute allergic response. Along with this, read about hypersensitivity reactions, all the types. They ask question about Ig3 subtype. So basically, what are Ig3 subtypes called? So they are basically allotypes. So read about what are isotypes, allotypes, idiotypes. Then, very very important topic, antigen processing and presentation. You should know which pathways are involved for MHC1 and in MHC2, antigen processing and presentation, and like what kind of antigens are processed in these uh, like processes. All this you should know. And also about the MSC molecules, you should know like uh, the kind of polypeptides they are made up of, what properties they show, like how are like they do so diverse in their function. Like these questions have been asked. I'll show you another one question which was asked from this topic. And also like which molecule they present the antigen to, what are the major what are the major like receptors for this recognition, co-receptors, that all should you know. Also the bonds which are interaction bonds and interaction that are formed between these molecules, all that you should know. Then lipid antigen presentation by which cell, antibody class switching, agranulated cells, complement protein related to opsonization. Then toll-like receptor types, basically whatever the TLRs are, 1, be 2, 3, 4 or 5 or whatever, and what they uh, recognize, that you should know, because I think they gave a match the following question regarding this. But you should know what they recognize. Some recognize double-stranded RNA, some recognize single-stranded RNA, some recognize CPG islands, some recognize bacterial flagellin, that you should know. Then immunotoxins, chimeric and humanized antibody. This was again asked twice, so you should know about this. Effect of endocytosis pathway mutation on antigen presentation. And again, analytical part. I don't remember the exact question, but it was quite easy if you have an idea about like antigen presentation and how endocytosis, like these pathways work. You will be able to solve this question. Again, phagocytic and non-phagocytic immune cells, rheumatoid arthritis treatment and involvement of Treg cells. So Treg cells is known for increasing peripheral tolerance and repressing autoimmunity. So it has been recognized as a potent like treatment for rheumatoid arthritis. So this read also about all the other types of T cells like Th1, Th2 and all like different type of T cells. Now, which B cell activated first, follicular, marginal or plasma? MSC independent cancer killing cell. I think the answer for this was NK cells. Process not involved in primary adaptive immune response. The options were affinity maturation, class switching, somatic hypermutation. And again, MHC class 1 and class 2 property, very very important. Now techniques part, again very important. They ask techniques to determine DNA protein interaction, and you should also know about DNA interaction techniques, DNA RNA interaction techniques, DNA protein, RNA protein, and even protein. Very very important. You should know what techniques are involved in determining these kind of interaction, because many questions can be formed from this topic. ELISA, again very important. Many questions, I think 3 or 4 questions were asked from this topic. Percentage SDS used for 250 to 450 kilodalton protein, so I think it was 10 or 12%. One question was asked from NMR. They basically gave a compound. They asked how many peaks, 1H NMR peaks are formed for that compound. Again, FSCS, fluorescent activated cell sorting. I think though 2 questions were asked from this topic. You can basically see also the question which is based on flow cytometry, as well as apoptosis related question, cell cycle related question. You should study from this topic. You can refer to Pathfinder Techniques book. It is quite sufficient, I'll say, to study these topics. But if you're not able to uh, like understand, you can refer to several other videos which are available on YouTube, or if you want me to make videos on any of these topics, you can let me know in the comments. So one question was asked from chromatophore. Frap related graph was given, and then you had to interpret the data. So basically they asked a lot of questions which are fact based, many analytical, and some you have to calculate, and some questions like they're quite easy, like they give you a graph, you just have to interpret it. So I find these kind of questions very easy and very doable. So practice these type of questions. You can practice questions from CSIR net part 2, because I think the level is around that only. The CSIR net part C questions are quite lengthy. NII doesn't give you like that lengthy questions, but I think the CSIR part section B questions are quite similar to this NII PhD exam. Now particle associated with X-ray diffraction, so this is electron. RT-PCR chirality determination by which method? So the answer was CT spectro. Band analysis with antigen and antibody. I don't remember the exact question, but it was easy. Cephadix use related to protein purification. Basically, it is used in chromatography, and like especially in ion exchange as well as ion exchange as well as gel filtration chromatography. So study chromatography as well, and what are the anionic and cationic exchanges which are involved, that you should know. Again, they ask question from protein and SDS. Protein gives one band in SDS but two in IEF. Probable reason. Blotting paper absorbed ink by which mechanism? Molecule present in chlorophyll. Pedigree analysis. Simple dihybrid cross. I think these two questions were from genetics and were quite easy. Now these three questions are from hormones part. You should study hormones because they tend to ask questions from hormones, and they have repeated this particular question in this year's monsoon paper as well. Which hormone controls postprandial glucose level? So the answer was GLP-1, or glucagon like protein 1. Again, 2-3 questions were from chemistry and 2-3 were from aptitude. I don't remember the exact question, but aptitude questions are quite easy. So if you have solved CSIR part A questions, you might be able to do these questions as well. Now 2024, I don't have a lot of questions, but so whatever I have mentioned those questions. You can refer to both this 2023 and 2024 questions, and again prepare whatever topics encompass these topics. Now, attaching C3b to a protein antigen will enhance antibody production or repress it. If you have an idea about complement system, you will be able to answer these questions. They might change the words or the framing of the sentence, but if you have concept clarity beforehand, this won't matter, because as you know, C3b is actually enhancing the antibody production. Actually C3d does it better, but C3b also does it. Now diversity of MHC is due to, the answer is polymorphism and polygyny. Antibody raised against a particular antigen detected by which method? Answer was ELISA. Question regarding phagocytosis. Also, along with phagocytosis, read about which motor protein is involved for this. So you can see actin, myosin, and all those related topics in which are involved in these kind of movements. Post-vaccination increase in which antibody? Adjuvant and hapten. Read about complete and incomplete adjuvant and haptens. You can also see the examples of different haptens that are commonly used. Then ELISpot assay, important. They have asked question twice. Basically, what this ELISpot assay is and what is the difference between ELISA and ELISpot assay. Cyclin and CDAs, you should know. They don't ask very in-depth questions from cell biology. So if you have a very basic idea about cell biology and the working mechanisms, you'll be able to do it. Now they gave a GFP graph, and I think they asked about the localization of certain protein according to that graph. You had to look at that graph and then identify the protein localization. So that question was quite easy. Now meiosis, DNA content and chromosome number. Basically, they asked they gave like diploid set of chromosomes, and they asked after meiosis, what will be the effect on DNA content and chromosome number. So meiosis, you should know mitosis as well. Then one question related to smooth ER feature. Now they ask was sarin gas target, which is acetylcholinesterase. Uh, Basically, they ask what is its target, and again there is another compound DIPF which also target the same enzyme, and basically they irreversibly inhibit this enzyme. Now NL is present in which region of protein? Along with this, read about also the other signaling sequences which are present in certain regions of protein. For example, KDL sequences which are present at I think the N terminal of AR resident protein. So that you should know. Beta oxidation, chymotrypsin again repeated. Lac, anti-epiopteron, cell volume calculation, which hormone controls postprandial glucose level, they have again repeated this question. Genetic mapping, very simple question was asked. Calculation from Gibbs free energy, activation energy graph. Quantitative PCR, low CT value means what? Which method used to determine terminal amino acids? Now again from vitamins, thiamine deficiency causes which disease? Most soluble in water, oxygen, carbon dioxide or ammonia. Antibody mediated killing of pathogen by which cell? T cell maturation steps in thymus. So T cell maturation is basically takes place in several steps. They are first double negative, they then double positive, single positive, likewise. So you should know about that process. Tuberculin test, hypersensitivity reaction, again hypersensitivity, to which hypersensitivity type reaction this tuberculin test belongs to. So you should have an idea about that. It belongs to type 4 hypersensitivity reaction. Non-phagocytic cells, enzyme replacement theory, therapy, and molecular interaction related questions. Molecular interaction related questions, basically what kind of bond is present between antigen and antibody, and again 2-3 questions from chemistry and 2-3 questions from aptitude that you will be able to solve. So take a screenshot from this video and try to read all these topics at least, and focus mainly on biochemistry, immunology and techniques. And if you have the time, read mole biology as well as cell biology and see at least the hormones part. If you do only these much of topics, you might be able to score very well in this upcoming exam. And if you like this video, give a thumbs up, and if you have any confusion regarding any further clarification that you need or any further topics that you need to study from me, do let me know in the comments and tell me whether this video was helpful to you. So take care everyone. Thank you.